Okay, so we are in part 3 of chapter 10, and we're looking at comparing meiosis and mitosis. Let's see, there are 2 divisions in meiosis, only 1 in mitosis. In meiosis, we do have synapse and crossover that occur during prophase 1, but we don't have any synapsing or crossing over in mitosis. Remember that synapse is when the homologous pairs find each other and pair up. That doesn't happen in mitosis. The homologous pairs exist, but they just don't. They're just randomly scattered in the cell. They don't find each other and pair up together. Let's see. It says the centromeres survive anaphase 1. What that means is that the duplicated chromosomes, in other words, the chromosomes that contain 2 sister chromatids joined at a centromere, they don't split in anaphase 1, but in mitosis they do divide, so the centromeres dissolve when that happens. In meiosis, the chromosome number is halved. In mitosis, it is preserved. The diploid number of chromosomes remains the same. So if you start out with 46 in the parent cell, the daughter cells will also have 46. Meiosis produces 4 daughter cells, whereas mitosis only produces 2. Meiosis produces daughter cells that are genetically different from the parent and genetically different from each other. In mitosis, it's kind of like cloning. The daughter cells are like each other and they're like the parent. Meiosis is only used for sexual reproduction, and mitosis is used for asexual reproduction and growth. So, there are 2 tables that compare the stages of meiosis 1 to mitosis and then meiosis 2 to mitosis. I told you that meiosis 2 and mitosis are exactly the same. If you read these, prophase 2 and prophase both say the same thing. They both say no pairing of chromosomes, no pairing of chromosomes. But metaphase 2, it says the haploid number of duplicated chromosomes lines up at the metaphase plate. In metaphase, the diploid number of duplicated chromosomes lines up at the metaphase plate. So if you can visualize this in your mind, in a human cell, what it would look like is, in metaphase 2, you would have 23 duplicated chromosomes. In other words, 23 chromosomes that have 2 sister chromatids attached at a centromere. So there would be 23 of these lined up. But in metaphase, it would be 46 of them lined up. Okay, so that is a difference as far as the number of chromosomes that we're dealing with, yes. In anaphase, the sister chromatids separate. In telophase, you end up with, in meiosis 2, you end up with 4 daughter cells, but in mitosis, only 2 daughter cells. So there are some differences, but as far as the process of meiosis 2, the same thing that happens in mitosis happens in meiosis 2. And this is a comparison of meiosis 1 and mitosis. Well, actually, it's showing you meiosis 1 and then the end of meiosis 2. All right, then we have this section on the life cycle. A life cycle of any organism is all the reproductive events that occur from one generation to the next generation. In plants undergo a process or a life cycle that we call alternation of generations, and that means plants can have a haploid multicellular individual that alternates with a diploid multicellular individual. This is something that we will learn more about in Bio 112, and it's just more than I want to go into today. But I do want to talk about the life cycle of animals, particularly the life cycle of humans. 1, animals are diploid and produce haploid gametes. So all of our body cells are diploid. In other words, they contain 46 chromosomes. The only cells that we have that are haploid are sperm and eggs, which we call gametes. The products of meiosis are always gametes. Meiosis always produces gametes, eggs or sperm. Now, the production of gametes is called gametogenesis. The production of sperm is called spermatogenesis. And at the end of spermatogenesis, all 4 of the cells become sperm. The production of eggs is called oogenesis. And by the way, spermatogenesis occurs in the testes of males. Oogenesis occurs in the eggs of females. But in the production of eggs during oogenesis, only one of the 4 eggs, one of the 4 cells will become the egg. The others will become polar bodies that cannot be fertilized. They're much smaller than the egg and they can't be fertilized. So sperm and egg are produced by meiosis during the processes of spermatogenesis and oogenesis. During fertilization, the sperm fuses with the egg and the resulting cell is the zygote. And the only thing I want to say here about oogenesis is it's just a little bit different, okay? So females begin producing oocytes, which are immature eggs. Females begin producing oocytes before birth. But the primary oocytes are going to start oogenesis. But then once the female has reached sexual maturity, once the female has entered puberty, those primary oocytes will undergo meiosis 1. And after meiosis 1, there'll be secondary oocytes. There'll be one secondary oocyte, and then the other cell is called a polar body. Now, the next thing that has to happen is this secondary oocyte has to undergo meiosis 2, but it doesn't complete meiosis 2. It stops at metaphase 2. And either one of 2 things are going to happen. Either a sperm will fertilize that secondary oocyte, and it will continue on and finish meiosis 2, or not. And if it's not fertilized, it will degenerate. Okay, so I just wanted to let you know, if the secondary oocyte is fertilized at metaphase 2, then it'll continue on to anaphase 2 and telophase 2 and cytokinesis, and it will form a secondary oocyte and another polar body. And the secondary oocyte will be the fertilized egg. This is just an illustration of the life cycle of humans, and everything up here in the top half is in blue. That means all the cells in the top half are diploid cells. So starting with the zygote or fertilized egg, undergoes mitosis. All of the cells in this child are going to be diploid. And then as the child grows into an adult, male or female, all the cells are going to be diploid except eggs and sperm. So in the sex cells, in the ovaries and the testes of these 2 individuals, meiosis is going to occur. In the female, it's going to be oogenesis, which will form an egg. And in the male, it's going to be spermatogenesis, which will form a sperm. And those 2 cells are gametes, and they're going to be haploid. But when they unite during fertilization, they'll be diploid again. So this is just an illustration of the life cycle of humans. Then you see an illustration here of spermatogenesis and oogenesis, which occur in the gonads of humans. And the last section that we have, I'm hoping I can finish it in this lecture, but the last section is on chromosomal disorders, abnormal numbers of chromosomes or missing pieces of chromosomes, different things like that, but chromosomal disorders. So when a species has their correct number of chromosomes, we say they're euploidy. That's the correct number of chromosomes in a species. So a human with 46 chromosomes is euploid. But a human who has a different number of chromosomes, like 45 or 47, they're aneuploid. The display of chromosomes, I've already shown you one of these, is called a karyotype. Now, aneuploidy, which is a difference in the normal chromosome number, always comes from a process called non-disjunction. And this is the failure of the chromosomes to separate when they're supposed to separate, either in anaphase 1 or anaphase 2. That can either lead to monosomy, which is only one of a particular type of chromosome, or trisomy, which is 3. Monosomy means that you're missing one chromosome. Trisomy means that you have an extra chromosome. Okay, so non-disjunction occurring in meiosis 1. Let's see what happens. Let's say that this cell has only one pair of chromosomes. They don't separate. So one of the daughter cells at the end of meiosis 1 has 2 chromosomes and one has none. The daughter cell over here on the left has an extra chromosome compared to what it's supposed to have. Over here is on the right side, you see what normally should have happened during meiosis 1. But anyway, since that daughter cell has an extra chromosome, when it divides the resulting gamete, which is in this case is going to be eggs, the eggs are going to have extra chromosomes. They should just have one red strand, one red chromatid here, but they've got 2. So when the sperm unites with the egg, instead of having 2 total chromosomes, one blue, one red, each of these 2 cells is going to have 3. So this would be what's called a trisomy. But the daughter cell that formed that didn't get any chromosomes is going to have 1 less chromosome than it should have. It should have had 1, it has 0. When the sperm fuses with it, it's going to have 1 chromosome rather than 2. And so that's going to be a monosomy. Okay, so this is what can happen if non-disjunction occurs during meiosis 1. Now let's look and see if it occurs, what happens when it occurs during meiosis 2. So the meiosis 1 process is normal. You start out with a pair of chromosomes and each daughter cell gets 1. That's what should have happened. Now, the daughter cell on the left undergoes meiosis 2 normally. And the sister chromatids separate. Each daughter cell gets 1 of the chromatids. But non-disjunction occurs in the daughter cell on the right. So one of the daughter cells that's produced here is going to have 2, which is an extra chromosome. And then one of them has 0, which is, 1 chromosome too few. So you end up with 2 of the 4 daughter cells normal, diploid, after fertilization. And then you end up with 2 of them that are abnormal, that have aneuploidy after fertilization. One has trisomy, an extra chromosome, and the other one has monosomy, which is missing chromosome. Okay, and here are the definitions of all of that. Trisomy occurs when an individual has 3 of a particular type of chromosome. Remember, everyone's supposed to have 2 of each type. So 3 is an extra chromosome. The most common autosomal trisomy, and autosomal means that this occurs, the most common trisomy that occurs within the first 22 pairs of chromosomes. It's called autosomal if it happens with one of the pairs 1 through pair 22. But when it happens on the sex chromosomes, then it would be a sex chromosomal trisomy. Okay, so on chromosome 21, if we have a trisomy or an extra chromosome there, we call it Down syndrome. Trisomy 21 is Down syndrome. The chance of a woman having a child with Down syndrome increases with her age. The longer the oocytes are stored, the greater the chances of non-disjunction occurring, basically because they are older. They're old and they don't work as well. Now, the characteristics of an individual with Down syndrome tend to be short stature, means they're not tall, eyelid fold, flat face, stubby fingers, and wide gap between first and second toes. This is what trisomy 21 looks like in a karyotype. Extra chromosome on pair 21. And then when we have trisomies and monosomies that happen in the sex chromosomes, which are pair 23, we can get some disorders as well. So Turner syndrome means you have, on pair number 23, you have 1 X, and then that O means nothing. So this is a female with a single X chromosome. The individual tends to be short with a broad chest, widely spaced nipples, normal intelligence, and usually we'll use hormone